Hello, my name is Ted Verhey, and I'm a postdoctoral fellow in Sir Anna Morrissey's lab at the University of Calgary. Our lab conducts genomics, single-cell and bulk transcriptomics, proteomics, and other omics profiling of brain tumors from surgical resections as well as in vivo models. Today I'm going to tell you about how we are using proteomics data to uncover new frontiers in brain tumor biology by looking at protein complexes. Glioblastoma is the most common and deadly brain tumor in adults, with a 5-year survival of 22%. Patients typically undergo maximal safe resection along with chemotherapy to extend life, but this always results in recurrence of the tumor. Why have traditional driver discovery and targeted treatment trials failed? The first reason is intratumoral heterogeneity, or the presence of many different clones within a single tumor, which enables adaptation to challenges including chemotherapy. The second reason is that these clones can be selected for over time so that the original clones need not exist in late-stage disease. The third reason for so few advances in treatment is that genomic alterations discovered to date have not had well-defined pathways of oncogenesis or functional outcomes. In order to address these issues, we have put together a proof of principle study to identify proteomic aberrations in space and time. To address spatial heterogeneity, we are obtaining multiple samples from different regions of each tumor. To address changes in time, we are sampling primary and all available recurrent tumors. To understand the functional outcomes of genomic alterations, we are using TMT proteomics technology to sensitively map out the proteomic phenotypes of each sample. We are beginning with a trial cohort of 4 patients, a total of 32 samples, and 2 replicates per sample for a total of 64 proteomic profiles. Many other studies have employed proteomics methods to uncover alterations at the protein level, post-transcriptional regulations such as alternative splicing and proteolysis, and post-translational modifications. In cancer, proteomics has also allowed for the discovery of neopeptides, which are of interest to targeted immunotherapies. More recently, proteomics has enabled the study of protein complexes, which may be dysregulated in cancer. Discoveries of protein complex dysregulation in cancer would lead to a novel class of anti-cancer drugs that stabilize or destabilize protein-protein complexes. Our goal today is to identify the extent to which we can identify protein complexes in our cohort using the correlation of member proteins and their peptides. To begin our analytical pipeline, we have samples that are multiplexed for greater quantitative accuracy using tandem mass tags. We then analyze these using the state-of-the-art Orbitrap Fusion Lumos tribrid mass spectrometer. Peptides and proteins are identified using Sequest-HT and Percolator for post-processing. Peptides are quantified relative to a normal brain reference sample that is included on each TMT array. We remove peptides with greater than 40% missing values so that correlations are not too noisy. We then generate a correlation matrix of the peptides and analyze the correlated peptides for complexes using the Quorum database. As you can see, we're able to quantify about 10 to 12,000 proteins from each TMT array, which corresponds to about 1,000 protein-protein complexes. We first looked to see whether we could observe a correlation between peptides from proteins of the same complex. In gray, I've plotted the correlation of all peptides versus all other peptides across our cohort. You see a distribution roughly centered on 0. When you only look at the pairwise correlations within known protein-protein complexes, you see a significant enrichment for highly correlated peptides that is slightly strengthened when you filter for peptides that are unique to the complex. This shows that we can detect complexes in our cohort using peptide correlation data. Within protein complexes, there is a range of correlations between member proteins and peptides. For example, in some complexes, the mean pairwise correlation or coherence between all proteins and peptides is high. This means that the complex member proteins are highly correlated in the cohort, and this is evidence of a stable protein complex. Others are much lower, and this reflects the presence of correlated and uncorrelated groups of proteins or peptides within a quorum complex. In extreme cases, this is evidence of mutual exclusivity, where 2 complex partners are anti-correlated and are not associated together in the same complex. Here we're looking at the expression across our cohort of highly coherent complexes. So that's complexes with a coherence greater than 0.75. The expression data you're seeing is a log ratio relative to normal brain. What is striking is that the proteome is highly heterogeneous within and between patients, while replicates are closely clustered together. Some complexes are highly associated with each other, pointing to large processes such as transcriptional programs or infiltrating cells that differentiate tumor phenotypes from each other. Here is an example of a complex with low coherence. In this example, I'm showing all the peptides that are distinct to the PSA PCI SCG2 complex. This complex is expressed in the extracellular space at different sites in the body. The color scale shows the correlation between peptides, and you can see groups of correlated and anti-correlated peptides. Along the left side, I've shown the annotations for the proteins and uniproteids that contain these peptides. So you can see that these patterns could be explained by anti-correlated protein abundances, specifically of serpina-5 and SCG2. Despite belonging to the same complex, we observe that these two proteins are anti-correlated, meaning that in our two samples, these complexes are likely dissociated. Here's another example of mutual exclusivity in a complex. The SCAMP complex genes are highly expressed in the brain, but SCAMP2 and 3 are correlated with each other, while at the same time being anti-correlated with SCAMP1, implying that the complex never includes all three members together, at least in our cohort. This complex, which is involved in trafficking proteins to the surface, may be relevant for neuronal biology in glioblastoma. In conclusion, we've shown that quorum complexes can be identified and characterized using pairwise peptide correlations in our cohort, which has spatial-temporal sampling in a smaller number of patients. Spatial-temporal sampling of tumors reveals heterogeneity of protein complex expression. And what we'll do in the future is to look at our expanded spatial-temporal cohort to explore the link between complex membership and other clinical features. We'd also like to validate dysregulation of complexes using a larger cohort, such as the NCI CPTAC glioblastoma discovery study. Finally, I'd like to thank Dr. Morrissey for initiating this project, as well as our lab for their support. We also acknowledge Canadian and local funding bodies, as well as collaborators at the University of Calgary and the PC Cancer Agency. Thank you for listening today.